So next one is Joseph Priestley. He had conducted experiment to demonstrate that the significance of air in the mechanism of photosynthetic process. So here he had taken a jar and a candle and a mouse or mice. Here he kept this candle and mouse inside the closed jar. So he had lightened the candle. After some time, so the candle was put off and the mouse was also died. So what happened is whatever the oxygen which was available inside that apparatus or the jar was being exhausted by the candle and the mouse. So that means for burning of the candle it requires oxygen, and the candle was burning till the oxygen was available, and once that oxygen had been exhausted, the candle was put off. And mouse was also of course we know that animals require oxygen for their survival. So till the oxygen was available inside that apparatus, the mouse was surviving, but once the oxygen had been exhausted, the mouse was also been died. The same experiment was conducted by using mint plant also. So inside the jar he kept an mint plant along with the mice and the candle. So this is a mint plant. So the candle was lit and of course the mouse was also been there. So here when the mint plant was kept inside this one, the mice was or mouse was surviving and candle was also burning. That means for burning of this candle, as we said that they require some gas, and for survival of the mouse also they require some gas. So that is being provided by the mint plant, and whatever the damage which is caused by this candle caused to the mouse and the candle was compensated by the mint plant. So that shows that mint plant is, or we can say that here the plants release the gases which are being damaged by the candle and the animals like a mouse. So this shows that some sort of useful gas is released by the plants which help in burning of the candle and also survival of the mouse. And oxygen was discovered by Joseph Priestley. Engine house, Jan engine house. The foul gas which is been released by the mouse and that candle been compensated by the mint plant, that is they are helping in the purification of that atmosphere. That was demonstrated by engine house scientist. The plants release oxygen in presence of sunlight. So this was demonstrated by using a hydrilla plant. Here they had taken an apparatus with a small beaker. This beaker is filled with water, and in this water the hydrilla plant is grown or hydrilla plant is taken. And this apparatus, the hydrilla plant, is being covered with a inverted funnel like structure. This also been filled with water molecules. So this entire apparatus have been kept under the sunlight. They had observed that small air bubbles are being observed or present in the tube like structure of the funnel, so which have been released from the hydrilla plant in presence of sunlight. So the air bubbles represent the gas vacuoles which contains oxygen in it. So this shows that the plants will release oxygen in presence of sunlight. So this is the one other experiment shows that during the process of photosynthesis, oxygen is released by the plants. Okay, next one is Julius Sachs. He confirmed that the plants will produce glucose. The green parts will produce glucose that will be stored in the form of starch. And the green parts are present in particular structures. So those green structures are called as that is chlorophyll. So this chlorophyll, which is present in special structures, lamellated structures, which is called as chloroplasts. So this was confirmed by Julius Sachs, where glucose is stored in the form of starch molecule inside the plants. And next one is Engelmann had performed an experiment. So by using a prism, so when the light is passed through the prism, the illumination of light is done. This illuminated light from the prism is focused on an green algae, that is cladophora. That is a green algae, and he had provided aerobic atmosphere in around that one, and he had introduced a suspension of bacteria. All the bacteria accumulated near that aerobic region. So that shows that oxygen is released by the green algae when it is performing photosynthesis by using the light which is illuminated on it. So here majority of the bacteria are accumulated in blue and red regions of the spectrum. So this shows that whenever the light is illuminated on the green algae, that is cladophora, so that by using that light they will perform photosynthesis, and when they perform photosynthesis they will release oxygen. So that oxygen will be utilized by the aerobic bacteria. That's why majority of the bacteria are being observed in the regions of spectrum where high rate of photosynthesis is done, that is blue and the red regions of the spectrum. Then Van Neel had performed the experiment by using some bacteria, that is a green and purple bacteria. So he had used this purple and green bacteria to demonstrate that the oxygen which is released during the process of photosynthesis is from water molecule. So here in this one he had taken carbon dioxide molecules along with hydrogen sulfide. Instead of, actually we know that in the normal equation of photosynthesis, along with carbon dioxide the water molecules will be taken. So instead of that one, as this when he had done the experiment by using green and purple sulfur bacteria, so he had used hydrogen sulfide in place of water molecule, and they produced the glucose molecules along with water, and they had released sulfur instead of oxygen. This shows that the sulfur having released from the H2S, which is being replaced in place of water molecule in the normal photosynthetic process. So here based on this one, it have been demonstrated that the carbohydrates are being produced by using the carbon dioxide, which is being used as one of the substrate for photosynthetic process. So in the Van Neel experiment, the process where hydrogen being taken from the oxidizable compound which is producing the carbohydrate molecules. So it can be shown in other form, like it is one of the molecule here, CO2, in presence of light. It is converted into 2A plus CH2O, and H2O molecule will be released. So in this one, the carbohydrate molecule is formed here by using carbon dioxide, and hydrogen is being taken from the oxidizable compound here. So in this process, this shows that the carbon dioxide is being formed from, carbon dioxide is used in the formation of a glucose molecule or the carbohydrate, and the oxygen which have been released in the actual photosynthetic process of the normal plants, it is coming from water molecule but not from the carbon dioxide molecule. So here based on this one, the actual empirical formula, if you see here, CO2 plus H2O give rise to CH2O plus O2. So this is an empirical equation for photosynthetic process. Then the overall equation for photosynthesis is CO2 plus H2O, 6 molecules of carbon dioxide and 12 molecules of water molecule in presence of sunlight and chlorophyll molecule. So it will be converted into C6H12O6, that is glucose, plus 6 molecules of water will be released and 6 molecules of oxygen are released. So this is a basic equation related to the photosynthetic process, where the carbon dioxide is reduced to glucose molecule or carbohydrate, whereas water is oxidized to release oxygen. So we can say that here two reactions are taking place, that is oxidation and also reduction. So that's why we say that photosynthesis is an physico-chemical process where oxidative reduction is taking place. So here the two substrates are participating in the process. Out of that one substrate, that is carbon dioxide, is undergoing reduction to form carbohydrate, and other substrate, water molecule, is undergoing oxidation to produce oxygen molecule. So this is the way how this basic history related to the discovery of various steps involved in the photosynthesis was done by various groups of scientists.